All right. Hi, everybody. Hope you enjoyed your lunch. Okay. All right. So lab for today shouldn't be too onerous, I hope. And so it's actually not lab two. I don't know why that's in there still. Um, Essentially, the idea here is that we're going to be using uh, corn to both look at monohybrid and dihybrid crosses. So there's going to be one of each there. And also, we're going to use that to collect data from those corn or corn kernels, really, and use that to carry out a chi-square test, right? And so this is a really good kind of practical example and use of the chi-square test, right? So this is going to help you both understand how it's done, right? The actual mechanics of it, but also the reason why you use it and why certain things work the way it does. And so get to course content. Let's get back onto that while that changes. Yeah. Ba-dum-ba-dum. So in many ways, I'd consider this a kind of revision, really, for the exam, both for the Mendelian genetics part, but also for the chi-squared part as well, too. Right? That's kind of the aim behind this, right? Is to reinforce those ideas. And so really, what we have in Blackboard is a copy of the questions that you'd be answered if this were like a lab handout, right? So in here, well, that's kind of a funky looking thing. We have down here essentially two different crosses, right? So you're going to have for each cross, a monohybrid and a dihybrid cross, you're going to have a series of questions, one based on the cross and the second one based on your chi-squared analysis of it. And so the corn we're using, you'd actually have, like a corn cob in lab, but instead we're just going to use pictures, right? And you should be able, from these pictures, to determine the different phenotype combinations of each corn kernel. So essentially, a corn kernel is an F2 offspring of a cross between two heterozygotes, right? So this is exactly the same principles as we followed in monohybrid and dihybrid crosses. You'll actually see the cross kind of laid out here for you, right? Which will kind of give you a good answer as to the two phenotypes that you're looking at, which one's dominant, and the ratio that you should expect in the offspring, right? So when you look at a corn cob like this, you're actually looking at the F2 generation of a reciprocal cross between two F1 individuals. Let's make sure I've got my chat up. And this is Jordan. Go ahead.
Hi, sir. You had said something about corn, like each individual kernel, I guess is the right word, is an organism into itself, correct? Like each one of these things
Yeah,
is something.
that's kind of a slightly weird thing about looking at corn kernels. So even though you have a corn cob, like the bit in the middle, I guess, right, that you leave behind after you eat it, is the parent, but each kernel is an individual offspring of that parent as a result of fertilization. So each one of these counts as an individual kind of organism, I guess. And each one has a different phenotype combination depending on Mendelian principles and the genotype of the parents. So that's the only kind of tricky thing to think about, is that really you're looking at the kernels. The kernels are the offspring of that cross. Does that make sense?
Yes, gotcha. So given that it's a little bit more complicated than that, can we just keep, is it just going to be kept at like, not surface level, but basically just like, this is the baby, or this is the F1, this is F2, as opposed to being like, in relation to the questions on the test is more my question, I guess.
Right. So what you're going to be doing to, just trying to think through your question, make sure I answer it properly. So when you collect the data, you're going to be doing so from this individual, well, this collection of individuals here, right? So these kernels are the offspring of two heterozygotes crossed together, right?
Gotcha.
And so at each step, you really observe the individuals that are going to take part in the next generation, if that kind of makes sense. So here you care about the kernels, because those kernels are going to generate plants that are going to mate to generate these offspring, right? And these kernels, they're all heterozygotes as a result. And so they will be kind of grown into plants and fertilizing each other to generate these offspring. So yeah, don't get too hung up on the which part of which plant we're talking about thing, because I think that just confuses things.
Yeah, that's what I was worried. Because if I see a question related to this, I'm gonna overthink or maybe go down the wrong path because of the way corn and what is, I guess, the individual for corn as opposed to like en masse.
Right. So for the purposes of this lab, what you care about are the phenotypes of the individual kernels. So even though realistically that isn't, well, yeah, just care about the kernels. Keep my mouth shut on that one. Before I tie myself in further knots. It's a little bit weird, I have to say. I'm not a plant person at all in terms of science. I love growing them and eating them, but plants are a bit funky in many ways. And so it's best just to kind of skip over that and worry about the kernels themselves. So these kernels in this picture at the bottom are the F2 generation. These kernels in the middle part are the F1s, and these kernels at the top are the P generation. So these are the individuals you care about, and their phenotypes are what you care about. So that's what you'll be using to answer the questions in Blackboard. So essentially this is split up into 4 exercises, right? The first 2 exercises for a monohybrid cross, that'd be the first one and the second one. And then 2 exercises based on a dihybrid cross, right? So essentially they're the same kind of, like they're repeats. It's just the first 2 are about monohybrid cross, so you're going to be asked questions that refer to that. And the second set of 2 are all about the dihybrid cross. So even though the questions are the same, you're going to be answering them based on the information that you're given in this image rather than what you were given for the first 2. And so as you can see in this one, this is a relatively simple cross, right? We have 2 different phenotypes for one trait, and that trait is kernel color. You can tell which phenotype is dominant because what did the F1 generation always show the phenotype of? The dominant parent. Yeah, sorry, I should have left you more time. Hey, no worries, Marcus. Right, so you know that these are true breeding. right? Because that's a given for a monohybrid and dihybrid cross. So you know this is homozygous dominant, and this one's homozygous recessive. You'll get heterozygotes, which will show the phenotype of the dominant parent. And then for a monohybrid cross, because you're only dealing with one trait here, seed color, you should get a 3-to-1 ratio of dominant to recessive phenotypes. So really the questions are just about answering questions about that. So that's kind of, ooh. No, you can ask a question now, Rose. Now's a good time as any. What's up? Are you typing? So we don't care about sex is the answer. So this is a not quite understanding the question. We're always, if we have a dioecious sexual reproduction, then you should, all things being equal, get an equal proportion of males and females. But we don't care about whether these plants are male or female in this because we're not dealing with a sex-linked trait. And so when we care, I think what you're talking about in terms of the expected values. So if we do a monohybrid cross and we count the kernels in the F2 generation, the ones in the bottom here, and another tip as well, if they have even the slightest bit of purple in them, they're purple. It's always like an awkward bit with corn. But the expected values are based on the F2 phenotype ratio for a monohybrid cross in this case. Right, and so we know that's always a 3-to-1 ratio because that's what you'll get if you cross two heterozygotes together. And so the expected values are based on the ratio of phenotypes and how many kernels you count, right? So for example, our expected, oops, spanner not working too well, digest my lunch, I guess. Our expected ratio of, what's that, purple to yellow in the F2 is 3-to-1, right? Our expected values, this is why it's a good kind of practice, right, for chi-squared. For purple equals the total number of kernels counted times the expected fraction. So if you count, I don't know, 300 total kernels, which you probably can't because I don't think there are actually 300 in there, but it's just a nice round number. Your expected for purple equals 300 times 0.75, which is, oh, should have chosen 400, shouldn't I? It's 150 plus 75, so that's 225. So that's how you get the expected values. You always times the total number of whatever's counted by the expected fraction for that particular phenotype. Oh, yeah, you know the weird thing about chi-square is that you should never round up until you're done. Because even though it's obviously weird to expect a fraction of an animal. It does matter because it's a theoretical amount. So it's still the expected amount, even though you're in reality, you'll never get that expected amount. You'll either get one higher or one lower, right? Even if your expectation was completely accurate. Yeah. So just to kind of continue the process along, the expected value for yellow is 300 times 0.25 or a quarter. So that's the first step of doing a chi-squared, is to first work out what you're expecting, right? And for this it's easy because Mendel's laws, right, and the monohybrid and dihybrid cross tell us, right, for an F2 generation of a monohybrid cross, you will always get a 3-to-1 ratio, right? So that's what we use to work out our expected. For a dihybrid cross, right, we have 4 phenotype combinations because we have 2 in each parent. We have these are round purple, these are wrinkled yellow, right? So we have 4 possible different combinations in our F2, and they're always going to be in a 9:3 to 3:1 ratio. So the math is a little bit more involved, but it's the same principles. If you count 60 of these kernels, then you'd expect the round smooth ones to be 9/16 of those 60, whatever that is. That makes sense? Just always got to remember to do it from the total, not from the value that you get for each individual phenotype combination, but from the total number that you count. So basically what you're being asked to do for this is, in the first exercise of the pair, essentially, right, you need to just answer some basic questions about the actual cross that you're doing, right? And you need to use the key up here, and use these words. I actually do go through this lab and just double check, because sometimes people put in like slightly different variations on this and Blackboard doesn't recognize that. So make sure you use the words here. Just make my life a little bit easier when I go through and grade them. Right, and so this also corresponds to here. So this is a purple and wrinkled, yellow and wrinkled, purple and smooth, yellow and smooth. For the first cross, you only care about one trait, which is color. We could have done this with the wrinkled ones. It's actually easier to do with color. And so in the actual Blackboard, you just need to go in through and answer some questions. And they're pretty straightforward, right? With a little bit of telling me stuff, right? Make sure you do pay attention to the questions, right? So genotype of the F1, I don't want to know its phenotype. I want to know its genotype, right? And then what you're going to be asked to do is essentially this question 7 through 11 are the second part of the handout, right? So we're no longer just looking at this just as a plain cross, but what you need to do is actually score, I'd say at least 30 kernels of this one and 50 of the dihybrid, just to give you decent enough numbers. And I want you to carry out a chi-squared cross to see whether or not your idea, right, about what is going on here is actually matched by the data that you collect. So count 30 of these and count whether or not, or score them, I guess is probably the better word, whether or not, in this case, they're purple or yellow. Again, slightest bit of purple equals purple. There shouldn't be any intermediates, but sometimes you get kind of like purpley looking kernels. Those are purple. If they're plain yellow, they're yellow. If there's a bit of purple in them, they're purple. It's just one of those weirdnesses of corn. Probably a transposon hoofing around in there causing problems, right? And once you have those data, then you need to carry out a chi-square analysis with them, right? Which is basically going through this process. And so really it's kind of like a stepwise run through of a chi-square test, right? I'm getting you to kind of figure out the expected values, right? Which is based on Mendelian principles and however many kernels you count. You don't have to count a round number, it could be 32. That's fine. Doesn't matter. You need to calculate a chi-squared value, right? Using the equation further up, or what we covered in class. Give me the degrees of freedom, p-value, and then a brief, did that do what you thought it would do, right? Did your observed data match your expected values? Which hopefully they will, right? Now, a quick point is that obviously I'm not going to do a chi-squared on everybody's individual data because that would just take forever, right? So I will be marking you based on, right, does that chi-squared value look kind of reasonable, right? Because of your expected values up here? Yes. Full marks, right? I'm not going to check whether it's right or wrong. I just want to make sure that you're doing it as you should be doing it. Degrees of freedom is pretty absolute, right? Is the p-value, does it match what you gave me for your chi-squared value? Right, so you can do so your expectations can be completely wrong. And as long as you carry out your chi-squared as you should do and put your chi-squared value here, you'll get the chi-squared value part right. Right, I'm not grading on whether your experimental design was right, just whether you did each bit of it right. Similarly, if you got a chi-squared value of 5,000, let's say, or something weird, and you put down the correct degrees of freedom, and you put down that it was highly significant, right? Less than 0.0001, you get that right. Even though it's kind of weird that you got that result, it's all consistent between the different steps, right? So that's what will get you, hopefully it'll all go smoothly, right? That's kind of like, that's the hope. But even if you get some weird and wacky data that don't really match your expectations, as long as you do the chi-square properly, like you get a value which looks like it should be about right, put the correct degrees of freedom down, and then you put down the matching p-value for that chi-square value in degrees of freedom, you get that question right. So I'm not grading you based on what you should get, because, I mean, you might get some stuff wrong or whatever, it doesn't really matter, right? What I'm grading you is, do you actually do the chi-square test properly? Even if you end up with a result which is like, how did that person get that? That's odd. Doesn't matter, I don't care. As long as you did the chi-square test properly and interpret it properly, right? So if you get a crazy significant p-value, then clearly your expectations were wrong, right? Or you did something weird when you counted them. Yeah, one or the other, right? So as long as everything's consistent, you get full marks. Whether it actually makes sense, not really that bothered, right? I just want to make sure that you're actually doing the process properly. That's what matters. Any questions so far? Hopefully I'm still there. I got a little note saying my Internet connection is unstable. Blame my wife for that. She's probably taking a call or something right now. Okay. And then the second one is really much a repeat of the first, but now you're doing a dihybrid cross, right? So now each has two different traits. Well, we're now looking at two different traits. Each parent has two different phenotypes for those traits, right? For seed color, we have, again, purple versus yellow, pardon me. But we also have round seeds versus wrinkled seeds, right? Is the second trait, like seed plumpness. And again, use this picture. So obviously you got to answer, you know, the questions as provided, and they're exactly the same in Blackboard. It's a 1-for-1, I think, match. Then use this picture, and although it's going to be a little tedious, it will be really straightforward. Just score these kernels, right? So that one there, that kind of shitty looking one, that's yellow and wrinkled. That one's purple and wrinkled, yellow and wrinkled, yellow and wrinkled, yellow, purple, so on and so forth, or purple and smooth, right? So score these kernels for the 4 different phenotype combinations, which are going to be round and purple and round, yellow and wrinkled, purple and wrinkled, and yellow and round, basically. And then use those and your knowledge of Mendelian crosses and dihybrid cross and so on. Oh, there you go. That's an even better picture. Use this picture. It's a lot clearer. That one's a little bit shitty. Carry out the chi-squared test for this. Yes, count the kernels in the corn pictures. So for the monohybrid cross dipper, this picture is just fine because it's kind of like low res, but it's just color. So color's easy. But I forgot that I put this one in here. And I'd recommend doing this on a computer because the color reproduction is going to be better than on a printout, resolution, that sort of stuff. Use this picture, and obviously don't go into the crappy bits at the ends, but try and score using this picture the phenotype combinations of each individual kernel for 50 to 60 kernels. So yeah, normally you'd actually physically be holding this in your hand going in the lab. Instead, you get a picture to look at. It's essentially the same thing. Does that help? Hopefully. So at the end of the day, it doesn't matter 2 hoots whether this is corn or gerbils or who knows what. Corn are easy because you get a whole bunch of F2s in a small space. If you wanted to count 60 gerbils, that would just be a crazy mess in the lab, right? But what we really care about is how well you understand Mendelian inheritance, right? Particularly the monohybrid and dihybrid crosses and their very rigid, formulaic kind of steps. And also using that knowledge to be able to carry out a chi-squared test, or 2 actually, and practice that so that you're well up on it for the exam this afternoon stroke tomorrow. Right, that's kind of those are the main outcomes. We just use corn because it's compact and easy to use. No other reason really. That's pretty much it. So again, in Blackboard, you're going to have the same thing for the second cross. You need to do the same deal. You need to write down stuff, and please do just follow the questions. Most people lose points on this lab by writing the wrong thing down. Like the phenotype is purple and smooth. So I'm not asking about the phenotype. I'm asking the genotype. Pardon me. and just go through and work out the chi-square test. Put down each different step, save and submit, and you're done. It's really fairly straightforward, but it's really good practice though. So the genotype will be the genotype, right? So if it's F1, it's going to be heterozygous. So how would you write down the genotype of a heterozygote for those two traits? Well, you'd probably want to use the nomenclature that they're using in here. It's actually written for you here. It's not hard. And the phenotypes will be in ratios, which are also given here. Maybe one day I'll block that off, just make it a little bit harder. But based on what you know about Mendelian inheritance in monohybrid and dihybrid crosses. And you'll need to use those ratios to work out the expected values. Because if you do a dihybrid cross, then you expect 9/16 of your F2 generation will be dominant phenotypes. Right, that's just what you would expect. Now you're never going to get exactly 9/16, so that's what the chi-squared test is for, is to determine whether the difference between what you observe, which is biology, and what you expect, which is math, are pretty much the same, or if they're really different. Valeria, I'd suggest doing 30 to 40 for this one, the monohybrid. Oh no, not that one. Sorry. This one, the monohybrid cross, and 50 to 60 of this one. And the reason is that for the dihybrid cross, you have 4 different combinations. And so you want to make sure you collect enough data that you're going to get a fairly reasonable number for the smallest combination, which will be the double recessive. What would that be? Yellow and wrinkled, I think. So you don't have to do each and every kernel. I mean, you can't really see each and every kernel. Just you need to do enough to be able to generate meaningful data for your chi-squared. And also because it'd be really boring to count every kernel. Now just a little bit like a sneaky tip. Hopefully that won't show up on the recording. Yeah, that's a lot of corn. You can actually buy these kernels. Obviously, you can buy them for kind of educational purposes. But there are some really, really cool varieties of corn particularly from Mexico and Central America, because we all just get the plain basic like jumbo yellow corn, but you can get some really, really cool varieties of corn and maize. Just the same as you can get like super cool varieties of bananas and potatoes in South America that you'd never see anywhere else in the world. But yeah, it's always kind of fun to think about too. Corny science. Oh, that's just terrible. Amazing. Oh no, I thought that was a spelling mistake. Your dad joking me to death here, people. Honestly. Do I have to end the Zoom? Is that what it would take? Yeah. Thought that was my job to tell terrible jokes. Amazing, honestly. Yeah, me too. So I don't have anything more to add for the lab. Not enough coffee and too much genetics, I think. Anybody have any questions for, say, last week's lab or for the exam that's going to be today and tomorrow?
How many questions?
40.
How many questions are going to be on the test?
40, 40.
I have a question. Well, it's kind of actually, I was going to ask if you could kind of work through a couple of problems with me. And I don't know if it could be like, if you want to do it until everyone's done or if they all want to hear. It was just the homework problems. I couldn't get the answers. So I don't really know what I was doing wrong in the chi-squared one. And then the number 9, where it was like the XY of the female flies, I think. So
All right, give me a second. I'll bring them up so I can see what you're looking at.
okay, thank you.
Is that which homework 1?
Yes, homework 1 is the one with the chi-squared one.
Okay, well, let's do that one first.
Okay.
So
And that one,
you should all by now be able to see your answers for that. I did answer a question for someone, but just for everybody, you'll see the answers the day after the due date closes. And that's the same for the exam as well. I might actually change that for a couple of extra days. Shoot. Just in case anyone's unable to make the exam for whatever reason, then I can extend it for them. Give me just a moment. Doesn't mean you'll be able to take it after the due date, but it just means that if anybody has any issues with like internet or whatever, I can delay the closing of the exam for those people. Where are we at? Yeah, so Dipper, if you ever have that issue, you should be able to pick the exam up again when you go back in. It's not a hard auto submission, I think is the best way of putting it. Yeah, so, but again, if anyone does have, I think it pauses, Rose. Again, and this is a difficult thing because I've never taken one of my exams online, right? So I don't actually know exactly how it works, but I think it pauses, and then when you go back into it, you can resume it. So I don't think that causes. I don't, to be honest, Dipper, I don't honestly truly know. But if you do, ooh, treats. If you do have any problems because of power outages or internet, whatever's, or, doesn't matter what, let me know and I can fix that for you. Okay. So as long as it's within that extended time for the exam, because as soon as the hammer comes down and that's the end of the availability of the exam or due date really, then everybody will be able to see the answers and the questions as well. So I can't then let you take it after that, if you see what I mean. That's why it's really important to kind of communicate any issues quickly. And I will take my laptop with me this weekend, even though I'm probably not going to want to look at it much, just in case anyone does have any issues. What was I thought? I totally lost the train of thought. Oh, I was going through the homeworks. Adam, I'm just going to answer your question quickly, and then I'll get back to Marissa's questions for the homeworks. So you measure based off a line at the top of the gel, which you draw in yourself. It could be the top of the picture, which is as good as any, or you can draw a line across the top of the gel and measure from that. As long as you measure each band in each lane from the same point, you're good. So it doesn't really matter where you measure from along the top, as long as you measure the same for every single band and every single well, or lane rather. Does that make sense? Cool. Yeah, because you're trying to measure distance, and if you're trying to calibrate your gel, then you have to do it the same for everyone. Rose. So no, actually on the assignment, the idea is that you create a standard curve for each gel and each marker, right? And you're using that curve to interpolate between markers, right? So you don't pick, if you have an unknown, you don't pick the base pair marker that it's closest to and kind of guess. You actually use the distance that that unknown has traveled, and then your graph, you find the distance, you go up to your standard curve, and then you read off horizontally the size of the band. So it's not an estimate at all, it's a calculated value based on that size by distance relationship, which is your line that you're going to draw. Okay, questions. So, ba-bum. All right, Marissa, which question was it? Was it the question 8?
Yes.
The goodness of fit?
Mm-hmm. Something that I was doing, I kept getting that 254 answer.
Okay, so the question is, imagine that you set up actually, you can all see the answers, right? I don't have to be shy about that because the homework's finished.
Yes, I have it pulled up already and everything, so.
Okay. Rose, I'm going to get back to your question a little bit. I just want to follow through on this question from Marissa first. Right, so this is the question. You're going to do a cross between, so essentially this is a dihybrid cross. And you know that because it's 2 doubly heterozygous F1s. So it's heterozygous for 2 different traits. And also because you have 4 different combinations in your F2 progeny, right, which you're only going to get if you have 2 different traits. And so, okay, so let's see, let's bring up the chat. You know what, let's, no, I'm going to do it in the chat. I was thinking about doing it on the whiteboard, but okay, Rose, don't let me forget to answer your question. I'm just going to start typing things in the chat to answer this. Okay, so first step is calculate expected values.
If you want, you can answer their stuff because I kind of want to see like a visual if you could.
Oh yeah, sure.
Thank you. That's
No
what I was
worries.
saying. We could wait until everyone's done because I know it may take a little bit longer.
Well, I mean, people can leave whenever they want. They're not forced to stay here, but I think this will benefit everybody because it's a good question to ask. So, oh, okay. So again, Rose, I'll draw this out for you. It's not a dumb question. It's one which makes a lot of sense when you know what you're talking about, I guess, right? As with everything, it's kind of like a mathematical thing. So essentially, let's get this off of here. Dum. So if you have a gel where your molecular weight marker and your unknown 1, let's say, oops, and looks something like this, right? And this is the question mark. And you know what this is. Let's say this is something like that, right? So you know that your unknown is somewhere between 400 and 500, or 600, sorry, but you don't actually know the exact value, right? Because it's an intermediate. And so what you do, and that's what the exercise is all about. I can't believe that dog chewed up my pen. Look at that. What a furry pest. All right, anyway, is, you draw a line across the top. It could be imaginary, could be an actual line. And then you measure the distance each one of these bands has traveled. Yeah. Then you plot, so you know the size, you've worked out the distance, now you can plot distance by size or size by distance, depending on your preference. You know, distance in centimeters or millimeters, doesn't actually really matter as long as you're consistent. And then your size in base pairs here, right? So this is going to be kind of odd to draw because this needs to be a log scale. So it's going to be a little bit weird. And it has to be a log scale, otherwise you'll get a curve. You won't get a straight line. Okay, so you measure this, that's going to give you this kind of distance. It's obviously an inverse relationship because small stuff travels further, right? So the bigger the thing, the smaller the distance. But you'll get something that looks like, how many we got? 5. Something like that. Give or take. Does that make sense, Rose? Okay. Now you draw your line of best fit. Oh, that isn't too bad. Between those points, and it's kind of got to fit all of the points as best as you can make it. And so now you have values in between, right? So not only do you have these 5 different values, but now you have every single value in between those 5 to calculate your unknown. Because that's what that line is there for. It's like the relationship between size and distance, basically. And so then you measure the distance that your unknown has moved. That gives you one piece of information, which it would be, say, here. Right, it's going to be a value in between these two, right? Because that's where it went to on the. You then draw a line up or vertically from that point on the x-axis and along, and you read out, I don't know what that would be. That'd probably be like 525, let's say. Whatever it says on the scale, which is going to be better than my hand-drawn one, and that's your actual size, or your calculated size, based on the line that you drew with this marker. Now, obviously, if you did this again with a different marker, then you may end up with a slightly different line and a slightly different calculated size. And so
I do have a quick question, if that's okay. So
yeah, for sure.
I'm looking at the Promega. So like in total, there's 13 markers, but we only know like for sure like 5 of them. Are we only using those 5 then, or are we estimating like all 13 of them?
Oh no, this is just because I didn't want to write out 13 because it would be really tedious. For the Promega one, you use which ones you can clearly identify from the picture and measure accurately. So you probably don't need to do all of them, but you should do as many as you can.
Gotcha. Great, thank you.
Yep. So getting back to Rose's question, you have 2 different markers. You have the Promega marker, and then on the other side of the gel, you have what's called the Edvotech marker. Names aren't really that important. They differ in the number. So Serena said there's 13 in the Promega one. I think there's 5 in the Edvotech one, right? But you need to draw 2 different graphs, one for each of those markers, one for the Promega one, and then you do the same for the Edvotech one. So Well, depending on which unknown you're looking at, there's different bands, right? So I think unknown 1 has 1 band, unknown 2 has 2 bands, and unknown 3 has 3 bands. And so you need to do this for each band. Sorry, noise from the kitchen. Each band of the unknown. So for unknown 1, you're going to give me 1 value, both in distance and the estimated size. For unknown 2, you're going to need to do this for the 2 different bands. And essentially you would get, say, 1 here and 1 here, for example. So you calculate the sizes of the 2 different bands. But essentially, everything you do for 1 marker, right? So the unknown 1, 2, 3, calculated with a ProMega marker, you need to do that again with the Edvotech marker. Because I want you to compare the 2, see which you think generates the most accurate estimates. And if you look on Blackboard, you're actually given the actual sizes as supplied by the manufacturer. Did that help? For everybody, not just Rose? Cool. Yeah, sometimes these things, particularly math stuff, if you're like me, doesn't always sink in like the first time, but sometimes a second go around kind of gives you the kind of twigs. It's like, oh, how about that? Kind of makes more sense. Okay. Hey, Valeria, I'm going to just make sure that I get rid of that in the chat before I post it up online, okay? I don't think it's an issue here. Actually, probably no one else can see it because it's Blackboard. But yeah, I try and remove things like that. Ugh. Oh, I probably shouldn't have changed position because I need to do that chi-squared test. Has anybody got any other questions about last week's lab before I go on to the chi-squared stuff? Oh. Sorry, I'll go back and have a look. It shouldn't be an issue as long as it's not on... The chat doesn't appear on the YouTube videos. The chat only is generated by Zoom separately. And that's what I post on Blackboard. But I do try and... Actually, it can be based on your own preference. If you're happy for me to leave it there, I'll leave it there. If you'd rather I didn't, then I'll take it off. I really don't mind. But any separate communications, I'd take off before I post them. Like if you're asking me individually, not the whole class. Okay, so let's get back to this. So I'm going to set this up so, tell you what, getting old sucks. It really does. Wouldn't recommend it for anybody. So let's get this in place. And then we'll work through the chi-square for Marissa. Okay, I'm just going to bring it up. That's why I'm not showing, so you can see this better. Okay, so we have dihybrid cross, F2 generation, our expected ratio, because we know what a F2 of a dihybrid cross is always 9 to 3 to 3 to 1 for the 4 different combinations. So our first step is expected values. You have to forgive my shorthand here. It just makes things a little easier. And so first we need our total, which is 272 plus 88 plus 107 plus 33. I'm going to need to use a calculator because my brain is about shot.
It's
So-
500.
Oh, awesome. Thank you, whoever mystery voice that was. I think that might
No
have been
problem.
Rissa.
I tried working through it a couple times. So like I do have some numbers, but I don't know. I just wasn't getting the answer.
No worries. Well, hopefully by going through it like this, and sorry, I can't actually see you right now because I don't have my Zoom up. Quite what often happens is when you go through it like this, you're like, oh, shoot, it was that thing. Like there'll be something that will jump out to you and you're like, crap, that's what I did wrong. And that's what we're kind of looking for, right? Because once you figure that out, the rest is easy. And so your expected values are going to be for your 9 to 3 to 3 to 1. Going to be 500 times, oh, I hate fractions. It's going to be 0.5. I'm not going to risk that. 9/16. There you go. Bit of a cop out there.
Okay, yeah, that's what I couldn't figure out was what to multiply it by. So that's where I was wrong, I think.
No worries. So that's where this is really kind of the most important step, really, of all, because this is where you take your hypothesis and use that to figure out what you should get if your hypothesis is correct. And so if your hypothesis is that you're looking at a Mendelian dihybrid cross F2 generation, then you would expect to get your results in a 9 to 3 to 3 to 1 ratio, roughly. So this is where we have to start. And so that's going to give you, give me a second. 500 times, oh, yeah, shift. 9 divided by 16 equals 300. And that's easy. Nope. All right, stop making leaps of faith there, Matt. 500 times 3. Sorry, it's a little slow. I'm just doing this one-handed. Oh, there you go. That was my mistake.
It's okay. I'm doing it alongside with you.
And don't round up, right? Until you get to the very, very end, if at all, right? Because even though obviously you're not going to get 3/4 of an individual, this is a mathematical deal. It's not like a practical one. This is what you expect based on kind of mathematical principles.
So why did you put 300 then if the, like you rounded up?
I think that was actually what my computer told me. So
I got 281.25.
Really? Perhaps I made a mistake there. Give me a second. Oh, yeah. Well, thanks for catching that. I must have typed something else with like, you should see me, I'm like all contoured up on my couch. There you go. Correct. Thanks for that. And then the final one is obviously 1 divided by 16 times 500. That's 31.25. So you'll need to keep like a handle of those because I need to rub this off. So you got those all written down?
Yes, I've got that down.
Cool. Okay, because I'm going to be asking you what they are in a second. So to be honest, this is usually the point at which most people mess up chi-squared analysis because the rest of it's fairly straightforward, right? But this is the most important part to get straight because otherwise everything else doesn't work.
Yeah, that's exactly what was happening. The numbers I have here were not what I had earlier.
That's okay. I mean, that's why we do this as a both the homework and the practice, right? And ideally, in an ideal world, I'd have more time to do this in class too, but sometimes we just have to get through what we have to get through. And so we just move that out. So the first one is going to be 272 minus, what is it, 281.25?
Yes.
Squared 281.25 plus 88 minus, what was it, 90 something?
93.75.
Yep. Squared. And then 107. Always get a little anxious doing these because I really hope that I got the answer right in the homework. Kind of like the doubter's curse of mathematics. And then 33 minus, is it 31?
31.25.
31.25 squared divided by 31.25. Okay, you can probably work that out quicker than I can. I'm just using the Windows 10 calculator on my computer. What does that give us?
Yeah, let me see. I'm just pulling it up here because I don't know why, but Blackboard kicks you out if the iPad, like the screen goes black, you know?
Oh,
so,
I'm sorry.
No, that's okay. I'm just writing it out here. I'm more like a visual person.
Mm-hmm. Oh, I wasn't actually, dang it. I just realized that my computer wasn't pointing in the right place. There you go. I'll leave that up there. I think you might be muffled, Marissa. I think you're speaking.
I'm just.
Oh, well, you're humming to yourself.
Yes, I am. Sorry.
That's okay. All right, what do we get?
I got 2.63.
And that is, I think, pretty close to, yeah, that's exactly what you should get. So that's actually one of the pluses of doing this stuff, is that as long as you follow the rules, right, and you're clear about the assumptions that you're making, your expectations, you'll always get the same answer. But that's the trick. Well, not the trick, I guess. The potential pitfall, right, is if you don't get part of that right, then it will get it completely wrong.
Yep, that's happened exactly last night.
That's okay. I mean, that's kind of the purpose of the homeworks, really. They're kind of a fairly low stakes kind of way of finding that stuff out. And I'd always much rather you find that out kind of in that context than in the middle of an exam. So degrees of freedom, pretty straightforward.
Yes, I got
Right.
that one. And the other ones too, using the chart and all that, that was...
Let's
those ones-
just bring that up. Oh, okay. Give me a second. Gonna move some things around. I just want to get back to...
One thing I noticed, I didn't know where to find the chi-squared with the, like, just the numbers though. Like, I did find the one where you start from the bottom and up.
Can you see what I'm looking at right now?
Oh, yep. Are you gonna add it there? Oh, perfect. Okay, thank you.
It's always been there.
So scroll down. Okay.
But I mean, really it's there for the exam today, but you can also use it obviously for the homework too. So we've got degrees of freedom of 3. So we're looking along this row. And what was it again? 2.63? I think
Yes.
the chi-square. So that's somewhere between 0.5 and 0.1 p-value. So you can't say exactly. You could work it out using this chart, which is a much more accurate way of doing it, but this is as good as we need, I think. It's basically the p-value is greater than 0.1. And as our significance level is 0.05, is that p-value significant or not significant? Question to you.
It is significant. If it's 0.05, or does it have to be less than 0.05 to be significant?
0.05 or less is significant.
Okay.
So to the right of this column, 0.05 gets ever more significant. Because in other words, the chance of you getting that result randomly, or probability of getting that result by chance, gets lower and lower and lower the further to the right of that table you get. So the less likely it is by chance, the more likely it is by something else.
So our data would not be significant.
Nope, because it is over on the left-hand side above 0.1. And really, once it gets above 0.05, it doesn't matter how far above it is, it's still not significant. This is the line in the sand, essentially. And so where we are. Yeah, there you go. So this is the value calculated from that chart, but you could also guess which one it is because it's between 0.1 and 0.5, right? And then, and typically if I ask you this question in the exam, I'll give you a range because I'm assuming you're going to use the table. And so that's kind of like the lowest common denominator, I guess. And so, yeah, it has to be, it's not significant, it's greater than 0.05. So the null hypothesis stands. Nothing's going on.
Okay, thank you. That really cleared that up. So,
oh, there's another one, right?
Yes, there's just one more. It was on homework 2 though, and it was number 9 with the yellow body in Drosophila. And then we had to set, if we had to set up the cross below. It was number 9.
Dum, dum, dum. Okay, so yellow body is X-linked recessive, so it's a sex-linked gene. and it's recessive. So that tells you that it will behave the classic kind of dominant recessive way, but it's going to depend on, because it's X-linked, which way round you do the cross, right? So you set the cross below what expected phenotype ratios, right? So you have a cross between, so the X's there and the large Y's represent the sex chromosomes. So the one on the left, the XX is female, and the one on the right is XY is male. Right. So, and this is part of this is just genetic nomenclature, right? And we'll cover this in much more depth when we get into the fly linkage lab next week. Yeah, next week. So it might make a little bit more sense by then. So anyway, what that's telling you is that the fly on the left is a female because it's XX, and one X chromosome has a wild type allele, plus allele, and one chromosome has a recessive Y allele, right? So that fly is heterozygous. Does that make sense?
Yes, that all makes sense right now. So, okay.
And the one
I
on
guess
the right,
I get confused because it's like on the top, you know, like it's
oh
not.
yeah, I understand. Okay. It's just kind of a way of writing this stuff, and you can write it in different ways too. But you know, this is just one of the ways. And so the other fly is a male, and for its sole X chromosome, it has a recessive allele. Right. And so this is the key point. So that fly, the male, will be yellow bodied because there's no second X chromosome that may contain a wild type allele to mask that recessive allele. It's essentially, it's not heterozygous, it's hemizygous, right? So it's half zygous, right? And that's the case for all sex-linked or X-linked traits. That's why you see color blindness more commonly in men, because one of the genes that controls that is on the X chromosome. So even if you had a recessive allele, you, Marissa, as an individual, most likely you would have a dominant allele on your other X chromosome, and so you would not have color blindness. Now, if I had that allele on my sole X chromosome, I would be colorblind because there's no other X chromosome there. It's just a Y. So that's the deal behind X-linked or sex-linked recessive traits.
That makes sense. So
yeah,
then
it's pretty cool.
You would answer the question just based off of knowing that information?
Right. So then what you have to do is a Punnett square. It's like the answer to everything inheritance-wise. So
do you mind doing that so we can see it?
Not at all. Let me finish this. 2.63. Okay.
Did the puppy get tired of being there already, or is he still there?
No, she's probably lying upside down, sleeping next to my daughter, who's on couch in the other room, would be my guess. Yeah, there you go. Feedback report from Sophie. She's a real cutie pie. It's actually astonishing, given how much she's gone through, what a kind of chilled out dog she is. Okay, so we have our heterozygous female, and we have our hemizygous male, right? And so, as always, you work out what gametes these individuals can produce. I didn't leave myself enough space here. Right, and I often just circle those because that reminds me that I'm talking about gametes rather than individuals. And then we have that. Right, so we're either going to have, for the females, they're going to, each gamete is going to have an X chromosome. One of them's going to have Y allele, the other one's going to have the dominant allele. For the males, one gamete is going to have a Y chromosome, so that will generate males in the next generation. The other one is going to have an X chromosome with a Y. And so you just add those together. So this will be that, and this will be so you will get, we're talking about phenotypes. Yes, we're talking about phenotypes. You'll get 1/4 yellow-bodied females, 1/4 normal or wild type females, 1/4 yellow-bodied males, 1/4 wild type males. As simple as that.
Okay, awesome. I think the words trip me up because I'm literally looking at it and I did that, but I just couldn't process, like, I don't know, I guess the actual answer. So, okay,
that's okay.
That makes sense then.
That's often like part of, unintentionally or intentionally, I'm not sure. The skills that we teach you is really kind of breaking down problems into smaller parts. So those, so you don't suffer from that kind of morass of information and like struggle to figure out what's what. And to be honest, I haven't seen this question for a long time. And so that's really just what I did. Obviously, I have the benefit of, doing this every semester. But the first step is always, what are we talking about as a trait? Are we talking about an X-linked recessive trait? So that's telling you something immediately, that this is going to be a gene that's on the X chromosome. Then wants phenotypic ratios, which is good, because genotypic ratios are going to be a bit weird. And then it tells you the genotypes of the two parents, right? And so one is heterozygous because it has a Y and a plus. The other one is hemizygous male because it just has an X with a Y allele. Obviously, if that male was actually a brown-bodied male, right, had a plus there instead, you'd have to do the Punnett square using those possible gametes, and you get a different answer, right? So you just really have to follow what you see. And then the key to all of these, right, because this is something that maybe you could work it out just in your head, but I'm not really someone that does that well. The key is just to write stuff down, right? And always you have a genotype. What are the gametes? The gametes are what combine with gametes from the other one to form the next generation, and that will give you your phenotype ratio. Then you simply just read it off. And so for questions like this, don't try and kind of figure it out en masse, right? You have to break it down into the individual parts and then just work through it, right? And in the simplest way possible, and then you'll always get the correct answer. It's when you try and kind of intuit something and you go, ooh, that looks like it'd be 50-50. That'd be wrong.
Yeah, I'm noticing I think what I need to do is also just look at like the verbiage and look it up, because it's going to help a lot with me knowing like how to process the question. So
yeah, well, also, I mean, my aim, which I usually meet, is to give you that verbiage and that language before you have to use it in an exam, because it's kind of rude otherwise, right? But we do use a very specific language, and you do need to know that, right? Because otherwise,
it's like Chinese.
It trips you up because you don't fully understand what the question is, and so you're not necessarily able to answer it properly. Right? Not because you don't necessarily know what the answer is, it's just because you can't understand what the question is, if you see what I mean.
Yeah, word problems have always like, I'm tripping on them. They always trip me up.
Yeah. Well,
okay, but thank you for explaining that.
Yeah, I mean, fully heads up, you will find questions like that in the exam, and you'll find questions that have a lot of, verbiage
Words.
in them and words and stuff. And so just take your time and just write down the key important parts and pull out what matters. Right. And then you can go through a process of elimination and go, well, it can't be that, or that, because, well, that's wrong. And it should reduce that kind of, I don't know. I always suffer from when, particularly if I've got a really messy workbench in the garage, like trying to find something. If there's a whole bunch of stuff around, I just can't see it. Not because I can't recognize it. It's just there's so much noise around.
Yeah, it's like coming all at you at once. So right, you're like, exactly. Okay. Nope, that's great advice. That's definitely what I'm going to do. Okay, thank you very much for explaining that.
No worries. It's a pleasure.
Other than that, I do not have any other questions. Thank you.
Cool. No worries. Is anybody else that's still with us? Got any questions about anything and everything? Now's your time. Dun, dun, dun. All right, as always, I'm available by email and stuff. So if you do have any questions, give me a shout. Try and do it before probably about 2 o'clock tomorrow would be ideal, because I'm going to be heading out in the afternoon with my family to go camping. I will take my laptop with me and I'll check it, occasionally over the weekend. Hopefully, I think they might have Wi-Fi. And just see if anybody has any issues. Okay, but if not, good luck with the exam. Be careful, take your time, and I'll see you next week.